Hi everyone, so here we are once again about to go through another past exam paper, and today it is the OCR A Paper 3 from June 2018. So what I always do is that I go through the paper, I show you what I would have written, I show you the mark scheme and all the examiner's notes, and therefore how many marks I would have got, and hopefully explaining the questions and why I thought of the things that I wrote the way I did. So I also carefully show you where I get every single mark, because the mark points in the way that they are listed can be easily numbered. So I put the tick and the number of the mark point right next to the word where I think I've actually secured the mark. So hopefully making all of that pretty clear. So let's get started. First of all, we've got an onion plant and we've got a table below about the root cells of onions, and we've got to place a tick in the appropriate boxes. Well, here is what I think makes sense. Roots, of course, do not contain chloroplasts. They do contain mitochondria. They don't contain 70S ribosomes in the cytoplasm because, of course, they are eukaryotes, so there would be 80S. They do not have pili because they are not bacteria. They are eukaryotes again. And it is true that they have cell walls. So here are the answers. Here's the mark scheme, and therefore I got the 2 marks. Here's a cross-section of the root of an onion plant, and you've got to identify the 2 tissues. So M is xylem. Xylem is really obvious because it's the great big tubes in a micrograph. And N, now be careful, because when I looked at N a bit quickly, I thought the line was continuing right to this point here. So I was nearly about to write endodermis or meristem. But I realized, looking at the picture carefully and on 100%, that it actually is pointing to here, which is the phloem. So, and I got the 2 marks. Notice that because they asked you the tissue name, you couldn't say the names of the cells because they're asking you the tissue name, and that is xylem and phloem. And now we have a little bit of genetics. Colour of onion bulbs determined by big A, little a, big B, little b. Big A produces red pigment. Big B inhibits big A. Little b allows red pigment to be shown, or red pigment to be produced, I should say that. So here are my little notes. Big A equals red. Little a equals white. Big B stops big A. Little b allows A. A white onion plant was cross-pollinated with a red onion plant. All 15 offspring had the genotype big A, big B, little b. Identify the following. Now, I started with the red onion plant because I got in my head very quickly that big A allowed red pigment or caused red pigment to be produced. But you had to have little b in order for that red pigment to happen. So red onion, the red onion plant had to be big A, big B, little b. Now, why is it homozygous for each of those 2 genes? Well, because we're told that all of the offspring are heterozygous for both genes. In other words, they're a bit of an old mix. So, and in order for that to happen, it had to be that the red onion was big A, big B, and therefore the white onion would be the opposite. Little a, big B. Notice that it couldn't be big A, little b, because otherwise you would have had to have this genotype for the A gene as big A, little a as well. And that would have produced the red pigment. Oh, sorry, no, it wouldn't have done because of the B, but you wouldn't have got all the offspring being big A, big B, little b. So hence that would be the genotype. And the phenotype of the offspring, of course, so they are red, but that red is inhibited and therefore they are white onions. State the type of gene interaction shown by the genes A and B. Well, it's epistasis. And if we're going to be precise, it's dominant epistasis because the B gene it's the dominant allele of the B gene that inhibits or masks the expression of the A gene. So dominant, oh, sorry, didn't do any of that. There we go. And this is epistasis and dominant. Notice that if you put recessive epistasis, you didn't get the mark. Whereas if you just put epistasis on its own, they gave you the benefit of the doubt. So in this situation, you had to be sure that your description of the epistasis was correct. Suggest how allele B inhibits the expression of allele A. Now, I must admit, I immediately thought of allele big A producing a functional enzyme that caused the production of the red pigment. And therefore, I had gene B being an inhibitor to that enzyme. Now, when I looked at the mark scheme, I actually only got one mark out of the two, because the way that the mark scheme went really wanted you to go down the route of allele, the protein for allele big B, capital B, produced a repressive transcription factor that bound to the promoter of the big A allele and therefore stopped the big A allele being transcribed, therefore translated, and therefore turned into a protein. Hope that kind of makes sense. So I only got one mark out of two on that one. ATP produced by a variety of ways. Describe the production of ATP by substrate level phosphorylation in different stages of respiration with reference to the number of ATP molecules produced. So here's my quick abbreviation to the examiner, because I really don't want to be writing out substrate level phosphorylation the whole time. And here is my answer. So I know that glycolysis produces 2 ATP net overall as a result of 4 ATP being made when triose phosphate is converted into pyruvate. And I realized when I got to the end of that sentence that I hadn't put in the detail that I knew that it's 2 triose phosphate being turned into 2 pyruvate molecules. So I chucked it in the brackets. And then I said, because it's asking about the numbers of ATP, 2 ATPs per glucose molecule, or I could have said 1 ATP molecule per turn of the Krebs cycle, because of course, if you think about it, if there's 2 pyruvate molecules being produced from 1 glucose, each of those pyruvate molecules is going to go into a Krebs cycle of its own, if you see what I mean. So from pyruvate onwards, all the numbers of things that are produced have to be times by 2. So 1 ATP molecule per Krebs cycle, 2 ATP molecules per molecule of glucose. And they're also made by substrate level phosphorylation when a 5-carbon compound, which you do not need to know the name of, is converted into the 4-carbon compound oxaloacetate, which you do need to know the name of. So here are the mark points, and here are the extra notes, and here are where I got the marks, or here is where I got the marks even. Notice that this note here allowed me to award myself mark point 1 and mark point 2 within the first sentence. And then I got another mark for actually spelling that bit out. I would never have guessed the mark scheme gave you so many marks for so many details, if I'm honest. I didn't know where the marks were, and therefore I put in the detail. Now, you may not believe me, and fair enough, but when I create this PowerPoint, I don't look at what I'm copying and pasting, and I deliberately write what I would write in the answer before I look at the mark scheme and the examiner's notes. So just saying how I do this. Oh, HSA, thank you for the question about whether I'm allowed to go through the 2020 papers. No, I'm not. As a responsible teacher, I would not go through any secure papers on a public medium. Teachers in schools would rightly want to string me up if I did so, because those are the papers that teachers in schools absolutely rely on as being secure for tests within schools to enable them to properly assess where students are at. So no, I'm afraid not. I won't do anything more recent than 2018. I may well go back, but 2019 and 2020 are both still secure papers at the moment. A little bit later on, I'll do 2019. Okay, so back to this. So this is where I've got mark point 4. So 1, 2, 3, 4, 1 ATP produced per Krebs cycle, but I'm allowed to say 2 ATP per glucose. Okay, when a 5-carbon is converted into a 4-carbon compound, oxaloacetate. So I actually ended up with 4 marks, but obviously the tariff for this answer is 4. I wouldn't have got over 4. Okay, 2, you're given a whole load of food tests for 3 tissues, A, B, and C. Use evidence in table 2 to identify which tissue, A, B, or C, is phloem. Have a think about what phloem does, and which tissue is liver. Have a think what kind of tissue liver is, and no, just think about what it is. So the most obvious to me, looking through here, is this result here. This is saying that it is negative for starch. These both are showing the presence of starch. Animal cells never ever have starch in them. So A is definitely the liver, because it's the only one that was negative for the starch test. Which one is phloem was more tricky, because here am I thinking, okay, phloem is it transports sucrose. Sucrose is a non-reducing sugar. So I'm expecting a positive test for the non-reducing sugar and a negative one for the reducing sugar. This is the reducing sugar and this is the non-reducing sugar. So, but blow me down, I've got positives for both. So here I am choosing which one I think is the best. Now, my logic is that it is tissue B is the phloem, because it shows the most non-reducing sugar in the sample. And so that is what I put as my answer. Has the highest concentration of non-reducing sugars, i.e. sucrose. Red, CF is notation for as compared with. Orange in C, so here am I comparing these two here. And A is the only tissue not to contain starch. So I got now I got mark point 1 and I got mark point 3. I just know I would never have thought of making a comment on these. I know it's 3 marks. I just wouldn't have made the comment. So, okay, cells can use fatty acids instead of carbohydrates as respiratory substrates. A process called beta oxidation, which you have never heard of and are not required to know about, is used to break down fatty acids into acetyl coenzyme A for use in respiration. Acetyl coenzyme A is obviously that substance that is funneled into the Krebs cycle in the mitochondria for aerobic respiration. Using information in figure 2, calculate the percentage of carbon atoms in the fatty acid that are able to enter the Krebs cycle. Well, this requires you to know that there are 2 carbons in the acetyl part of acetyl coenzyme A, which is funneled into the Krebs cycle. And now we're looking at the fatty acid. Well, the fatty acid has got 14, so 15, 16 carbons. So the percentage of carbon atoms is 2 over 16 times by 100, which equals 12.5%. And I got the mark. And these notes very helpfully tell you exactly how to do the calculation. And now we go on to the next calculation. The percentage of carbon atoms that a reaction makes available for use in the Krebs cycle is described as the efficiency of the reaction. Calculate the efficiency of the link reaction and use your answer to say whether it is more or less efficient than the reaction with all the fatty acids. Well, the link reaction, there are 3 carbons that at the start of the link reaction, pyruvate, and 2 carbon atoms in the acetyl part of the acetyl coenzyme A that get funneled into the Krebs cycle. So it's 2 out of 3 times by 100, 67% efficient. We've already worked out that the fatty acid one is 12.5% efficient. So clearly the Krebs cycle is more efficient. And I got the mark. Notice that they are very hot on rounding. Your last figure cannot be 6 without a dot over the top of it to indicate that it's recurring. You had to round it up appropriately. And of course, you could have an error carried forward if you'd got your calculation in the previous question part wrong. Figure 2 shows the role of coenzyme A in beta oxidation. Okay, right. Suggest a role for coenzymes other than coenzyme A in beta oxidation. And you might immediately think, I know nothing else about coenzymes in beta oxidation. However, let me just point out to you that they're indicating hydrogen atoms transferred here. And I would hope that that would give you a clue about something that you could write about. Because even though you don't know about coenzymes in the fatty acid oxidation, you do know about coenzymes that carry hydrogen atoms in respiration. And those, of course, are NAD and FAD. So I put NAD and FAD carry hydrogen atoms when reduced, i.e. NADH or NADH plus or FADH2, which dissociate to provide the electron transport chain with electrons, and I really should have put a comma here, plus hydrogen ions. Because I wasn't sure for OCR where the mark point would be. Would it be for carrying hydrogen atoms like the diagram showed me, or would they actually require more detail, and would they really want me to notice that it's actually carrying the electrons that are used in the electron transport chain? I didn't know, so I put everything in. And as it was, they just wanted to know about the hydrogen atoms. Notice you are not allowed to say hydrogen ions or hydrogen molecules because that is not true biologically or chemically. And so it carries the hydrogen atoms. Just a quick note, I have a website which has 99% of the syllabus of both OCRA and AQA on it, with me doing teaching videos for every part of those syllabuses. If you want to subscribe to both years, so if you're about to go into year 13 and you need both years' content, it's going to cost you 50 quid to subscribe for the year. That is a flipping bargain, can I just point out? Because private tuition is about £50 an hour, and there are hundreds of hours of videos. Well, yeah, probably. Obviously, watch them on double speed. Don't ever use the whole time. That would be daft. If you just need the AS videos, if you're just about to start year 12, then it's £25 for the year, and you can earn your money back. There's the crucial thing, because this is me wanting to make A-level biology accessible to all students, regardless of their family income. So if you recommend these on to other students, you get 10% commission on what they buy. And there's a freebie, so go and have a look anyway, even if you don't buy anything, or subscribe. Okay, oh, and I should be saying, oh, please like and subscribe to my YouTube channel. I hate saying that so much. I said it in a stupid voice, but you know, actually anything to get the word out there that I'm here to help A-level biology students. Spread the word however you're flipping like, I don't care. Anyway, back to question 3, photosynthesis. So here we have a student investigating how both light intensity and temperature affect the rate of photosynthesis for good old Canadian pondweed, Elodea. The rate of photosynthesis was measured by counting the number of bubbles produced by each plant per minute. Identify the anomalous result in table 3 and explain how this result could be confirmed as an anomaly. So I was running down these. So you would expect that at each temperature, as the light intensity increased, that the number of bubbles increased. And, you know, that is what happens. So I went back and looked at the numbers again, because I'm spotting an anomaly. There should be something way out. And I look at them carefully, and here we have it going up and it's plateauing. Oh, this one is hardly going up at all. It's not really plateauing there, and this isn't really plateauing either. I think it's 28. So there's the ring round it. So I said that it is the figure 28 at 40 degrees C at 32 light intensity. And the way that you would confirm it as an anomaly is to repeat it and to make sure that, you know, or to see whether you got the same result again. And if necessary, if you really thought it was still dodgy, repeat it again and check your methodology, check you're doing everything correctly, because it could be a, I've now forgotten the type of error that it is, if it just is because of, oh my goodness, can't remember the type of error that it is when you just do your method wrong. It'll come back to me. Hey ho, let's move on. So here is the mark scheme. Here are the examiner's notes, and here are my marks. Actually, 2 really quite easy marks that was. Okay, describe how the student could improve their experimental methods and the presentation of their data. So here I've put a little tiny, you know, synopsis of what we were told. The asterisk here tells you that it is one of those leveled quality of communication questions. So you need to write clearly and logically. So I immediately kind of went, okay, I'm going to deal with the experimental method first, and then I'm going to deal with the presentation. So experimental method. So let me just put it up because I can't remember offhand. So this, oh yes, of course. First one, really obvious to me, should have used a gas syringe rather than counting the number of bubbles, because then you have a measurement of the volume of gas rather than just number of bubbles. Bubbles can, of course, be completely different sizes. They should have also repeated each test at least twice. In other words, at least 3 trials for each light intensity and temperature, and therefore I'm being able to calculate a mean for the mean oxygen volume. And I said that they could have also used more temperatures up to 70 degrees and more light intensities. They should have controlled the mass. So there was me thinking, okay, what about controls? You know, okay, they are varying both the temperature and the light intensity. What could they control? The only thing I could really obviously think of was the mass of the Eladia. In terms of in data presentation, they should have presented the data as a graph. That seemed most obvious. And I probably should have said line graph, actually. Should I have said line graph? Not with only 4 points for each light intensity. And so that trends could be identified. And possibly to use separate tables per temperature. So one table for 25 degrees C, one for 40.5, and one for 70. And also notice right at the end, units for light intensity weren't given, and they should have been. Okay, so here is your classic descriptor of the answer that they're looking for. They wanted, for the 5 to 6, they wanted detailed descriptions. And the points that I'm about to put up here indicate what they mean by detailed, which is really useful. Level 2, marks 3 and 4, provide correct descriptions of improvements to both presentation and experimental methods, of both. And 1 and 2, provides a correct description of an improvement to both of the categories. And then depending on how well it's been communicated, you either got the higher or the lower mark within the band. So here are the possible scientific points. Please notice these ticks do not mean marks. This is me just kind of tracking what I've hit in terms of the scientific points they were possibly looking for. So detailed descriptions, and notice that I've also done a little tick. These bold parts are what they were looking for, the kinds of things they were looking for in terms of detailed description. The normal writing is just the point. The bold is the detail. So notice here, only here have I written detailed. I've written a detailed point here. I've written a detailed point here. And I wrote a detailed point here. I'm not convinced that I really expressed myself well enough about having smaller and more consistent intervals. Not sure about that. And I also completely forgot to mention how I should have added sodium hydrogen carbonate to the water to provide a controlled source of carbon dioxide. So I missed that. So I don't know, I wasn't absolutely sure whether I would have got 6, and I ended up giving myself 4. I would have either got 4 or 6 because I think I would have won the communication mark under both categories, both levels. Okay, photosynthesis occurs in 2 stages, light dependent, light independent. Explain why temperature has a greater effect on the rate of the light independent stage. I always have to remember that light independent means the one that isn't involving light. I have to think consciously about that every single time. I gave the examiner this as my abbreviation, light independent stage, to avoid me writing it out. And I said, because the light independent stage is an enzyme controlled step. For example, Rubisco, I didn't know where the marks were and whether I had to name the enzyme that you are expected to know about in the light independent stage. As temperature increases, and then I must admit, I kind of wrote, well, what else do I write for a second mark? And then I remember my own little advice to me, my own little advice to myself and to students. If you're answering an explain answer And you know the headline answer. In this case, light-independent stage is enzyme controlled. And I've still got other mark points to win. If you can't think of anything else obvious, and even with this Rubisco, I wasn't sure that that was going to get me the second mark, then go into describing your biology in more detail. So here I am now, or you could say I'm doing a further explanation of what it means about the light-independent reaction, light-independent stage, being enzyme controlled. As temperature increases, the kinetic energy of the enzymes and the substrates increase, leading to increased enzyme substrate complexes. I was very happy when I started writing this sentence and I realized I was going to end up talking about enzyme substrate complexes. Classic bit of enzyme terminology to get into an enzyme answer. So here's the mark scheme. So I got this mark here. Oh, here we go. There's, and that's higher temperature will increase the kinetic energy or increase the number of, and I could have even gone on to say about the enzyme denaturation, but to be quite frank, I completely ran out of space. So I think I would have stopped if this had been me in the exam. Probably worth noting though, that relatively my writing here is way bigger relatively than it would be on an actual exam paper. I may well have gone on an actual exam paper to say, and when the temperature goes above the optimum, enzymes are denatured. Right, okay, complete the following passage. Cells are removed from the meristem tissue in axial buds or root tips. I kind of thought axial buds, well, that's the shoot tips, or no, they're not, they're, you know, but they're on the shoot. So I did root tips. The tissue sample that is removed is called the explant. Ethanol can be used to sterilize, and the mass of cells is called a callus. So there's the standard mark scheme, but the notes allowed me to have root. Notice they didn't allow the word callose because callose is a completely different term to callus. A-gamma globulinemia, great, and Vichy syndrome are both genetic diseases. A-glob is what I'm going to call it now, results in a lack of mature B lymphocytes in a person's blood. Suggest and explain one symptom. Well, they're going to get infections more easily. So I don't know why I didn't put that have got infections more easily, because that would be a symptom. They would have had a weakened immune system because of fewer antibodies to cause pathogen destruction and fewer BM lymphocytes, memory B cells, to provide immunity. Just notice that it is the antibodies that actually cause you to recover from an infection, and it is the memory cells that give you immunity if you get infected again. Just can't miss the opportunity to clarify that. So here are the notes. So I got mark points 1 and mark points 2, actually right up at the start of the answer. What conclusions can you draw about the location and the nature of the allele responsible? As soon as it said location, I'm kind of going, they're either going to want me to talk about different chromosomes or the X chromosome, sex linkage. And the nature of the allele, here am I immediately kind of going, they're going to want me to say whether it's recessive or dominant. So looking at this family tree, I'm kind of going, looks like it's sex related, sex linked to me, not sex related, that would not be appropriate. Sex linked to me because the allele, because the gene, sorry, let me get my terminology right, because the gene is on the X chromosome. So, and here I might put allele. I should have put gene. I must check that the marks, I got them, they weren't worried about that in the mark scheme. Alleles located on the X chromosome, it's sex linked because all of the sufferers are male. Yeah, there isn't a single female sufferer anywhere. The allele is recessive because the male sufferers, 3, 7, and 8, have parents, neither of which have the disease. because that is my definition. So notice that I used my definition of sex-linked. Sex-linked means that the gene is on the X chromosome. Recessive means that a child can have the condition or disease even when neither of the parents have got it, because it means that both of those parents are heterozygous. And then I just went on to say, because I knew it's sex-linked, okay, I'm going to nail this. I'm going to make sure that they know that I know that the male offspring inherited their the recessive allele from their mother, because the mother is heterozygous. Okay, so actually I was right with the marks. So I actually ended up getting, I think, all the marks on this answer. 1, 2, 3, 4, 5, 6. Yes, I did. Obviously, tariff is only 4, but I hit all the points, which was nice. Veitch syndrome is a genetic disease that shows a recessive inheritance pattern. Notice this is nothing about it being sex-linked. This is just recessive. The allele responsible for Veitch syndrome is found on chromosome 18. They're telling you it's not sex-linked. Chromosome 18 is an autosome. Only chromosome pair 23 are the sex chromosomes. 2 carriers of Veitch syndrome have 6 children. Calculate how many of the 6 children you would expect to have Veitch syndrome. Well, I was doing this in my head and kind of going, so recessive, I've noticed that. So here we go. Big B, little v, sorry about the dodgy lettering. But I had to make my lowercase v a very different to an uppercase v if I'm making it denote the different alleles. So here is your classic heterozygous cross. You'd have a 3-to-1 phenotype offspring, 25% chance of each child having Veitch. And notice a 50% chance of a child carrying Veitch syndrome, being a carrier for Veitch syndrome. So, but of course, to have Veitch, you've got a 0.25% chance. 0.25 times 6 is 1, sorry, I should have said, is 1.5. I don't know why that didn't come up. Is 1.5. But you can't have 1.5 children. And so my logic was, okay, I've got to round it up to 2. So that's what I did. Carriers, 0.5 times 6. This is nice and easy. This is 3. Here's the mark scheme. Syndrome, you could have put 1 or 2, fairly reasonably. Carriers is 3. Oh, do you know? No, I didn't get this mark. This wasn't 1/2. This is 1, or I had to write 1 or 2. I've just realized I didn't get this mark. All right, because I needed to write 1 or 2. Do not allow 1.5. I missed that because I just thought about rounding up. The daughter of these parents, and so the daughter of these parents and a male carrier of Veitch syndrome have a child. So the male carrier, that means the male carrier is big V, little v. A daughter could have been any of these 3 genotypes. Calculate the probability of a child between these 2 having Veitch syndrome, i.e. being little v, little v. You've got to go through all of them. This was a load of work for 1 mark. So this is how I did it. I realized, I went through each of them. Homozygous dominant with the male carrier, there's absolutely 0 chance of a child having Veitch syndrome, because you cannot get homozygous recessive genotype with these 2 parental genotypes. So 0 chance. Now we go to the heterozygous cross, the father being big V, little v, the mother being big V, little v. And of course, there is a 25% chance, or sorry, 25% or 0.25 chance of the child having Vichy. However, notice that there is the chance of a child, a daughter of these parents. There's a 0.5% chance overall of the daughter being a carrier, higher than, well, you can't get higher than 0, it doesn't matter how much you times it by. And then we come to the daughter, little v, with the male carrier. And here, there is a 0.5% chance. So here we've got a 0.5% chance, here we've got a 0.5% chance. This one is out of the offing, which is not included in the calculation because it is 0. So now the question is what do we do with these numbers? Do we add the chances, the probability? I should have said probability. I just realised here I should have said probability. Probability, chance is not a good word to have used. Yeah, probably. Sorry, that is bad use of terminology on my part. There is a 0.5% probability. So do you add the probabilities or do you times them? Well, if we'd added them, the probability would have ended up being 1. In other words, there would have been a certainty of a child with Vichy being born, of the child, their first child having, and that, my logic tells me that that isn't correct. So times them and you get 0.25, which just intuitively feels more sensible to me. And that was the right answer. I struggled to get, I mean, I vaguely got my head around this, but this is what I would have done with regards to my maths. But my goodness, it would have been really easy not to have included this and therefore to have done 0.25 times 5 and ended up with 0.125 as the answer. Anyway, okay, moving on. DNA profiling can be used to analyze the risk of inheriting conditions. To produce a DNA profile, DNA first needs to be purified. Explain why a protease enzyme is used in the DNA purification. Well, that's because DNA associates, when it is packaging itself up in the cell, it associates with histones, which are proteins. So if you need to purify the DNA, you want to get rid of the histones. In other words, digest them away. So I said so that you could digest the histone proteins so that they can be removed. So, and there you go, proteins with DNA or histones. Ignore digests. Oh, ignore digest breaks down enzymes. Right, yes, fair enough, yes. Okay, so I got that mark. All right. They can be, DNA samples can be analysed using the polymerase chain reaction. In theory, how many fragments of DNA may be present after 12 cycles? Assume that you're starting with 1. So here is my single DNA fragment. With 1 PCR cycle, we get 2. With 2 PCR cycles, we get 4. Every cycle doubles the number of strands, of the number of DNA fragments. So therefore, you've got to do 12 times 2s. In other words, 2 to the power 12, which equals 4,096. Represent your answer as a log 10 value. Well, this is where I went to my calculator, because then I pressed 10 to the power X button on my calculator, and that gave me the number of 4,096 as 10 to the power of a particular number. The X is 3.61, So it's 10 to the 3.61. And that was the right answer. Notice you got 1 mark if you got as far as saying 4,096. Suggest why the figure that you calculated may not be achieved in practice. Hi, Lavinia, nice to see you. Stormy Doctor, I don't know quite what the wire is about. Maybe it's because we're going through this so flipping fast, but I hope it's impressive, whatever. Anyway, let's keep going. Suggest why the figure may not be achieved in practice. You are now having to, because I nearly just wrote, oh, because PCR isn't 100% efficient. Well, then I thought, they've just told me that in the flipping question. Why might it not be achieved in practice? I'm simply repeating the question. That is never going to get me a mark at A level. So here am I now thinking about, right, the PCR process. So the PCR process, you know, heat is applied to denature to separate the DNA strands. Then, you know, the heat is lowered to allow a primer to anneal. And the heat is then upped again to allow the polymerase, the DNA polymerase, to do its lengthening of the fragment. So you could have kind of picked any of those details. Maybe that not all the DNA natured, or not all, some of the DNA. So I went with some of the, not all the primers annealed, or that some separated DNA fragments may rejoin before the primer anneals. That's what I went for, but they basically wanted a detail from PCR that would explain why it wasn't 100% efficient. Yeah, so any of those. State the name of the enzyme used to synthesize the new DNA strands. Well, that is DNA polymerase. And if you wanted to be precise, it's Taq polymerase, because it has come from Thermus aquaticus, because it is not denatured at 95 degrees C, and therefore PCR is a cyclical process. It wouldn't be cyclical if the DNA polymerase got denatured on the first cycle, would it? So there we go, that's why. Okay, DNA fragments are separated to produce a DNA profile using electrophoresis. This guy wrote a description, described 2 changes that you would make to the student's procedure. So I was reading through this, and I must admit, I was thinking, oh, is it cathode? And I thought, no, cathode's right. Voltage will be passed through the gel for 1 minute. And it was only when I got to this point that I thought, 1 minute? It's not going to take, it's going to take longer than a minute. So, but I must admit, I'd got through 2 of the lines out of 5 before I thought I'd spotted my first possible mistake. The gel will then be placed in purified water. Here's another one. It isn't going to be purified water, it's going to be buffer solution. And we'll be able to see the banding pattern of each DNA sample. And to be quite frank, it isn't quite as simple as that. So 2 changes, and the ones that I thought were the most obvious was that the time was too short because it wouldn't allow for the full separation of all those DNA fragments, and that, oh, sorry, I've just realized I haven't done that, and that buffer would be used instead of purified water because buffer has got ions in it, and therefore it can carry the current, the electric current. Purified water certainly won't. Those 2 to me were the most obvious ones. Notice here and explain. Don't just describe the changes. Oh, I'd use a longer time, and I'd use buffer instead of purified water, because you haven't finished. This is only 2 marks. You have to nail the explanation for each thing to get each one of the marks. Quite hard marks to get. So here is where I got the marks. So I started off with 4, 1, 2, 3, 4. I started off down here, longer than a minute to ensure the separation, and then about the buffer solution as well. Accurate analysis. Anahita, I love the spelling correction. Thank you very much. I'm not a miss, but I'm really appreciating the fact that you're being polite. I totally answer to miss. I think nearly every teacher does, don't they? Okay, so right, blurdy blur. Spruce pine plant is given the binomial name Pinus glabra. Place a tick in the box next to the species most closely related to Pinus glabra. Well, I think rather obviously it is Pinus. Oh, this was a bit of a nasty one. I didn't even notice. I'm going to be really honest. I didn't even notice these two because this one was so obvious to me because it's got the same genus name. Don't be put off by the fact that just because it's got the same species name, they've got to be. It's got to be in the same genus before they start using the same species. These are in different genuses. Can't be genii, can it? Anyway, moving on. That is correct. Explain why Pinus glabra and humans, Homo sapiens, are classified in the same domain, but different kingdoms. So they're in the same domain because they're both in eukarya, because their cells contain nuclei and membrane-bound organelles. Okay, they are in different kingdoms. Pinus is in plant, Homo sapiens is in animals, because Pinus cells have cellulose cell walls, whereas Homo sapiens cells don't. This is using your knowledge of what is the difference between the 5 kingdoms, which you learnt about at AS, and you had to be able to nail that. Then the scientists sampled a species of trees present in 2 different habitats containing Pinus glabra. The results are shown. So I just ran my eyes down these habitat A and habitat B, and I just immediately noticed that habitat A has got a lower species richness than habitat B because it hasn't got either of these 2 species in it, whereas habitat B is only missing 1 species. So immediately habitat A has got a lower species richness. Okay. Oh, do you know, I should have said right at the beginning, if you've got any questions, because I'm going through this really quite quickly, do feel free to type away, and I'll happily come back to them and try and answer and explain things more clearly if I need to. And I got these 2 marks. So they gave you examples here of why, or a description. Oh, I actually hit both of those plants. Description of difference between plants and animals, and that to me was the most obvious one. Okay, using Simpson's index of diversity, the scientists calculate the biodiversity of habitat A as being 0.71. Use the formula given to calculate the biodiversity of habitat B. Show your working. Now, this is one of those, if you're not familiar enough with this, this formula, it can look a bit scary. The thing to decipher this, big N is always the total number of individuals of all species, and little n is the number of individuals in a particular species. So these numbers, both of these numbers, are little n. Okay, and you've got to total up these numbers to get big N. So if I'm doing habitat B, I need to do a total of all of these to get big N, and that is 104. So now we can come to this horrible bit of the formula. So what I have done is little n, 60, divided by big N, 104, squared on each of these lines here. This number here is 60 over 104 squared. This is 10 over 104 squared, etc., etc. And notice that once you have done that, you can then sum them. Oh, and here's another little hint. If you have a horrible formula like this with loads of brackets and squares and all the rest of it, start in the middle and work out. If you can suss out what n, little n over big N is, then you can square it. Then this means the sum of. And then if you've summed it all up, you're then taking that sum away from 1. So there's my other little tip. Start from the innards and work out. So here is the sum of little n over big N squared, which is 0.389. So now to get D, I've got to take that away from 1, which equals 6.11. So if this is 0.61, this means it has a lower biodiversity index than habitat A, and therefore A is the habitat with the greater biodiversity. Oh, I've just realised this was left up there. Sorry about that. So I got both the marks for that. Habitat B was situated beside a lake, showed evidence of succession. Right, okay. And plan to investigate how the biodiversity changed from the edge of the lake to the other side of habitat B. So in other words, I'm kind of visualizing going from the edge of the lake and away from the lake to the other side of habitat B. State the collective name of the animal and plant populations that are present at the end of primary succession. Well, that's the climax community. Suggest how the scientists could achieve the following during their investigation. Sample all stages of succession in the habitat. Well, that would be by doing a transect. And I could have just written doing a transect, but I kind of thought, sample all stages of succession. Okay, let's really nail this. I would do an uninterrupted belt transect. Okay, sorry, climax community, I got that. Belt transect. Oh, I should have put, okay. So to minimize, let's just go through, to minimize sampling bias. Now this was a tricky one because you kind of went to, okay, we're doing a transect to minimize sampling bias. Now bias is always talking about doing something randomly, but you can't place quadrats randomly if you're doing a transect. So does it make sense that actually you're eliminating bias by pre-deciding where the quadrats go on that transect? You could also have said, because my other possible answer was to say that you did many transects and positioned them randomly around the lake. Or you could do, because here I'm envisaging a lake, and come on, not all the habitats going away from the lake are going to be the same, are they? You possibly could have said uses stratified sampling as well. We're really going for it. But anyway, that was the answer that I went for. Here is the marks. Oh, sample insect biodiversity. You could say anything that talked about you sampling insects. So I got the marks for those. A scientist also measured primary production in both the woodland and lake habitats. And I can't, oh, this was the units one. So measured primary production. Primary production is mass divided by the area, excuse me, the area of land that the primary production is being assessed on. But then you have to remember that there is also a time factor, because how much, you know, I don't know, how much grass grows, well, it's obviously got to be per unit of time. So there has to be a unit of mass, there has to be a unit of area, and there has to be a unit of time in the woodland one. For the lake one, doesn't make sense for it to be area, it's got to be volume of water. So it has to be mass by volume of water by time. So here are my answers. kilograms, and these are just what I thought was the most sensible, kilograms per meter square per year and kilograms per meter cubed per year. So, and you can see all of the very various permutations that you could have written. But the crucial thing was that you had to talk about, I must say, actually, you didn't have to talk about kilograms. It was the most obvious unit to me. You absolutely could have talked about units of energy, because primary production, we're talking about measuring biomass, which is why I thought of kilograms, but biomass is the measure of the energy in an ecosystem. So you could have absolutely have used energy units at the start there as well. So hence all the many and varied permutations that you were allowed to have. Okay, moving on, I think. Oh, that's me showing I've got the marks. Yeah, okay. Process of ultrafiltration in the kidney shares similarities with the formation of tissue fluid. Describe the similarities and differences between ultrafiltration and the formation of tissue fluid. Deep breath time. Personally, this is an asterisk question. This is going to be leveled. It's going to be about your written communication, how clearly you can write it, how well you can lay it out. Is it logical? Is it using terminology, etc.? So I would absolutely, because I noticed that it is just talking about describing, just had to describe them, but we had to talk about similarities and differences. So to me, the obvious thing was to actually write 2 subheadings. Oh, and I gave the examiner my abbreviations for these. So I allowed myself more space because I would have relatively had way more space on the exam paper. So similarities, both filter the blood through capillary endothelium. Both involve capillaries. They filter out everything except blood cells and large proteins. They filter out they produce a filtrate containing water, glucose, amino acids, ions, and urea. Differences. Okay, some of these were okay, some of these were not. Ultrafiltration occurs between the capillary and the Bowman's capsule epithelium. Tissue fluid formation occurs in every capillary bed, all right, because the tissue fluid bathes the cells. Ultrafiltration occurs under very high pressure, as the efferent arterial diameter is less than the afferent arterial diameter. Tissue fluid formation occurs under less pressure, although it is still high, because capillaries have a smaller diameter than arterioles. Here am I using A-level standard knowledge. I'm not going beyond what you're expected to know. Ultrafiltration, capillaries between, this is the only thing that possibly I could be being called a little bit of a clever clogs. Not something that is particularly pointed out in many syllabuses, by many teachers maybe. Capillaries between the afferent and efferent capillaries. Oh, I've just written the wrong word. I should have written arterioles. So I wouldn't have got any marks for saying this at all because I wrote the wrong term. Here am I wanting to point out that the capillaries really unusually occur between 2 arterioles in the Bowman's capsule in the kidney, whereas for tissue fluid formation, the capillaries are always between arterioles and venules. Okay, so what I did here, because I must admit, I thought, good grief, I'm not. This was really quite tricky, I thought. So I looked at the scientific points before I looked at this bit, probably wrongly. And so I did a quick tick, a little tick. Notice my ticks are not marks. Little tick of which scientific points I think I hit. So I want to just talk through these. So I got this one, both occur in capillaries. but large molecules and proteins and cells remain in the blood, and that small molecules are filtered out, glucose, amino acids, ions, etc. I also hit the high hydrostatic pressure in both, even though I talked about it as a difference, because I really want to emphasize how much higher the pressure was in the Bowman's capsule. It's why it's called ultrafiltration. Ultra meaning lots of, or under high pressure, extreme circumstances. I did not include, I would not have considered including either this one or this one or this one, neither any of these dotted ones, because to me this is talking about the whole process of tissue fluid balance, or what happens after ultrafiltration. I absolutely focused on the filtration that happens in the Bowman's capsule and the formation of the tissue fluid, not what happens after the tissue fluid has been formed in terms of water being reabsorbed back into the capillaries and all that kind of stuff. There's no way I would have included those. I wouldn't have included, this confuses me slightly. I would have gone with this, that both include basement membranes, because capillaries have a basement membrane around them. However, I happen to know that the basement membrane between the Bowman's capsule epithelium and the capillary endothelium is fused, which kind of makes sense of this thing, of this sentence here, but it doesn't make sense of why it's only one named label in tissue fluid, because you would have thought that this would be endothelium, basement membrane, epithelium, and this would be endothelium, basement membrane. So I don't quite know, if I'm really honest, I'm just holding my hands up, I don't quite know how both of those are in the mark scheme. Don't quite get that. These two, this one I was very sad that I missed, and this one I was a little bit sad, but I wasn't going to kill myself. Yeah, sorry, that's a horrible expression. I wasn't going to beat myself up about it. See, that's not so nice either, is it? Anyway, and then I looked at this bit, and I suddenly realised I really didn't have too much to worry about, because level 1 correctly describes similarities or differences. Well, I'm all right on that. Level 2 correctly describes a similarity and a difference, one for each. And level 3 talks about correctly describing similarities and differences between the processes. Clearly, I have done multiples. And I think that my terminology, I think my layout, the logic of it would have got me the highest mark. So I did give myself a 6 for that. Okay, we're getting close to the end, guys. The GFR provides an indication of the health of kidneys. Measure the volume of blood that's filtered by the kidneys every minute. It can be estimated by monitoring the blood concentration of creatinine. Don't just go with it. Suggest 2 characteristics of a patient that must be taken into account when using this measurement. Actually, I've thought of one that's more obvious. One, I should have written recent diet, because your diet depends on how much creatine there will be in the blood. But another really obvious one, and I think almost more obvious than age, or it might seem more obvious than the diet one to you, is that actually how much exercise has somebody done? Because potentially the exercise load would have made a big difference. So here is the mark scheme, and here am I. I've got mark point 1, and I got mark point 2 and mark point 7 and 8. Notice again, explain why each characteristic must be considered. You've got to give some extra information why what you have suggested. Here are the suggestions: diet and age. Why would they make a difference? I actually think this one would have also been, I think that would have been an obvious one to have written as well, given that you learn about phosphocreatine in the respiration topic. If kidney damage is suspected, is likely to be tested for albumin. Explain why the presence of albumin indicates kidney damage. Well, that means it's got through the ultrafiltration process in the Bowman's capsule, and that means that there has been damage to either the Bowman's capsule or the glomerulus. So indicates damage to Bowman's capsule, capillaries. I should have written glomerulus there. I don't know why I didn't, as albumin should not be present in the filtrate. Fortunately, well, it says idea that large proteins should remain in the blood. So I've said that they shouldn't be in the filtrate, which is the same thing. And there we are, finished. So if you have any questions on that, do start typing now. But if not, I will love you and leave you until next Tuesday. I will be putting up some kind of decision as to what I'm going to be doing next Tuesday. I might well go through another exam paper because that seems to be quite useful at the moment, given that my website covers all of the content. So hopefully that has been useful, and I will see you this time next week, or a bit hour early, on Tuesday to go through another exam paper. Okay, have a good week, guys. Bye-bye.